Right. Good afternoon, or perhaps good morning or good evening, depending on what time of the day you are watching this lecture. So, happy Thursday. It is currently Thursday, March the 26th, and welcome to our first online lecture for Bio185. I hope you all are having a good day currently right now. Like I said, it is in the afternoon. It's pretty warm. It's in the 70s. I just took the dog around the block because I have been trying to film this lecture all day and bad things keep happening. I keep messing up, or programs keep failing, or currently in the background, if you hear my neighbor mowing the lawn, they have been mowing the lawn now for about 2 hours, and it's really starting to irritate me because they started and then they stopped, and I thought I was okay to try to film this again, but nevertheless, they apparently decided to take an hour break, and now they're back at it again. So I apologize if you hear that in the background. I hope that you all have been staying safe, and I hope that you all are ready for your first lecture. So I'm going to try to keep things as authentic as I can to how we have typically been doing it in person, and I ask that you are patient with me and know and understand that I will be patient with you as well, as this is a big transition for all of us, and clearly I am struggling, and I suspect that many of you will also be struggling, but know that we are in this together and we will get through it. So I don't know about you, but it certainly feels as if our first day discussion of aerobic respiration was both just yesterday but also eons ago, and my brain is really foggy and I don't remember much of what we talked about. And so what we're going to do to kind of refresh our minds and get ourselves back into what we were doing before our lives were turned upside down is we're going to start with a review or a recap, and then we're going to give a general overview of aerobic respiration. So what happens as a whole? And then we're going to get into stages 1 through 4. We're going to talk about what goes in, what comes out, and for each of those stages, we're going to talk about an overview. And our overarching question that we're working on answering here is just this idea of how does a cell break down glucose to obtain energy? Seems really simple, right? So this morning when I think my husband probably had chocolate for breakfast, because that's usually what happens. Chocolate has a lot of sugar. How did his body take that glucose, that sugar? How did it break it down to obtain energy? So we kind of take it for granted that our bodies do this every day. It's a pretty complicated process, but we are going to break it down into the steps. So we're going to start with a brainstorm question or a brainstorm problem. So again, my goal is to try to keep these online lectures as similar to what a typical in-person lecture would be. And that means we're going to have brainstorm questions. We're going to have practice short answer and practice multiple choice questions throughout the lecture. And while I will not be there in person to walk around the room and to hear you brainstorming your ideas with the people around you, I encourage you to pause the video and to work through these problems on your own. And even more so, I encourage you to maybe watch the lecture with a friend from class, virtually, of course. We want to maintain social distancing. And then maybe work off of them and try to make it as authentic as possible. So what I'm going to have you do is pause the lecture and to think about the key points from topics 13, which was an introduction to energy and metabolism, topic 14, which was enzymes, and then topic 15, so the introduction to this topic. And again, these were things we covered before break. So go ahead and pause the video and let's see what you come up with. All right, hopefully you've taken a couple minutes to brainstorm some ideas, or if you haven't brainstormed ideas, hopefully you at least looked back at our old topics and you looked back at those study guides that we made together as a class and were able to pull some of those pieces out. So the first thing that we're going to start with is we're going to start with energy. So when we first started our entire unit on metabolism, we started by defining energy as the capacity to cause change. And within energy, we have two types of energy. We have kinetic energy, which is the energy associated with motion, and then we have the energy that I would say that we're more interested in, and that is potential energy. And that's going to be energy that is stored. More specifically, the type of potential energy in this class that we're really interested in is chemical energy. And chemical energy is just a type of potential energy, and specifically, it's a type of energy that is stored in chemical bonds. So that's the first thing we started in this unit, was just an idea of what is energy. From there, I'm going to go ahead and put up a blank page here. From there, we moved into the idea of metabolism. So before break, I showed you this really crazy, complicated picture of the metabolic map and all of the chemical reactions that are happening in an organism. And we define metabolism as the sum total of all chemical processes in an organism. And we said that there are two types of metabolism. The first type of metabolism is catabolism. And this is taking larger molecules and breaking them down into smaller ones. And this type of metabolism is associated with release of energy. On the flip side, we have anabolism, or anabolic reactions, which are going to be reactions where we take simple molecules, so smaller molecules, we add in energy, and we build more complex molecules. So that's going to be like taking a bunch of amino acids, building a polypeptide from that. So we've got our peptide bonds or our covalent bonds that are holding together those amino acids. And all of those bonds have chemical energy or potential energy. So from there, we then had to think about, all right, well, how do we look at energy in biology? And how do we figure out, is a reaction going to be a catabolic reaction or an anabolic reaction, or is it even going to happen? And that takes us to Gibbs free energy. So when we talked about Gibbs free energy, we talked about it and defined it as being the proportion of a system's energy that can do work. In short, really Gibbs free energy looks at, will a reaction happen or not? And we're going to be looking at delta G, so the change in Gibbs free energy, and we'll draw some reaction coordinate diagrams here in a little bit to remind ourselves of what this means. But we can have delta G that's greater than 0, which is positive. And if we have a positive delta G, that tells us that the reaction is not favorable. If you have a delta G less than 0, or a negative delta G, this tells us that a reaction is favorable or spontaneous. And so again, we're going to take these pieces and we're going to combine them together. So before break, we were kind of adding on all of these pieces. So again, we're just kind of pulling out the key points here. So I'm going to put up another blank page. And after we defined Gibbs free energy, some of the things that we talked about were endergonic and exergonic reactions. So let's go ahead and draw both an endergonic reaction and an exergonic reaction. So let's go ahead and draw our X and Y axes. And throughout this topic, we're actually going to be drawing these reaction coordinate diagrams, so this will be a good review for that as well. Let's go ahead and label our x-axes with time and our y with G. So for an exergonic reaction, exergonic reactions are going to result and a release of energy. So we're going to start with our reactants having a higher potential energy, a release of energy, and then our products will have a lower potential energy. So let's go ahead and draw this in terms of catabolic and anabolic reactions. Oop. All right, so we've got our reactants and our products labeled here. And when we have a release of energy, we said that in terms of metabolism, the type of metabolism that deals with release of energy is catabolism. So this is a great example of what a reaction coordinate diagram for a catabolic reaction would look like. The other thing we have to throw up here is our labels. So if we take the potential energy of our products and subtract it from our reactants, we get our delta G. In this case, we would get a negative delta G. So this would be a favorable reaction. And that delta G, there's the amount of energy that would be released. The other thing that we can label and add on to here is what this little hump is, and that is activation energy. And activation energy is the amount of energy needed to start a chemical reaction. So again, we are starting with a larger macromolecule and breaking it down to its pieces. That larger macromolecule is held together by covalent bonds. We know that covalent bonds are the strongest of all the bonds. And so, of course, in order to break them, we have to have an input of energy that's going to allow us to do so. And then we're able to harness the energy in those bonds and to use that to do work. So now let's go ahead and flip over to the right side of our screen and let's talk about an endergonic reaction. An endergonic reaction is going to need an input of energy. And this is going to be an anabolic, or our anabolism for our metabolism. So this is going to be taking our reactants, and those are going to be built together into a larger macromolecule. So for an endergonic reaction, as compared to an exergonic, we have a lot more, or a higher activation energy. So that hump there, that is the entire amount of energy that is needed to start the chemical reaction. And just like with our exergonic reaction, the difference in that potential energy between the products and the reactants is our delta G. And in this case, our delta G is positive, which means that it is an unfavorable reaction. So one thing that we have to remind ourselves is anytime we have an endergonic reaction and an exergonic reaction, they are going to be coupled. So an endergonic reaction is not favorable, it's not spontaneous, it's going to require a big input of energy. So we're going to couple that to reactions that release energy. So we will see throughout this topic how the cell is able to do that. And while we're on the topic of endergonic and exergonic reactions, hopefully you're yelling at the screen, Dr. Hubbs, but what about enzymes? What do enzymes do? And so this is a great point to introduce and to remind ourselves of what enzymes do. So enzymes are catalysts. So that means that they are not used up in the reaction, and their entire role is to lower activation energy. So we talked about how enzymes can lower activation energy by having an active site that perfectly fits with the substrate. And so we talked about kind of the two models this idea of induced fit and the lock and key model, they can also, with the amino acids that are found in the active site, perhaps we need a really acidic environment. And so we find acidic amino acids there that are able to help lower that activation energy. So what we're going to do is on our left picture here for our exergonic reaction, we're going to draw what this would look like if an enzyme was present. And I'm going to use the same color, so I'm going to use this light pink. If an enzyme were present, that activation energy would be lowered. Everything else is the same. So keep in mind and recall from before break that enzymes do not alter delta G. They do not make a non-spontaneous reaction spontaneously spontaneous. That doesn't happen. The only thing that they do is they affect the activation energy. So they will decrease or lower the amount of input energy needed to start a chemical reaction. The lower the activation energy, the faster it will happen. So now that we've got those pieces and we're building on what we talked about before break, the next thing that we have to think about is, well, how do cells actually get work done? So we know that cells do work, and they do this with energy. So the question becomes, well, how do they do that? And in cells, all the time, right now, this is happening. We have lots of transfer of energy. And this is happening, like I said, all of the time. And there's two ways that we can have transfer of energy and two ways that we're going to talk about for this class. The first way that we mentioned is the transferring of phosphate groups between ADP and ATP. So we introduced the idea of kinases and phosphatases. So a kinase is an enzyme that transfers a phosphate group to a substrate, and a phosphatase is an enzyme that removes a phosphate group. So we introduced this idea of kinases and phosphatases, and we talked about how ATP, or adenosine triphosphate, has more potential energy than ADP. So when we take ATP and we break it down or we hydrolyze it, that's going to be an exergonic reaction, and we're going to be able to harness energy from that and then use it to do work in the cell. So we can actually go ahead and draw what that would look like. So let's draw our reaction coordinate diagrams. So here is ADP plus P going to ATP. So ATP has a higher potential energy. This is an endergonic reaction. We are taking that delta G, that energy is being stored in that chemical bond. On the flip side, for an exergonic reaction, we're going to have ATP going to ADP plus P. So again, we're going to have release of energy there. And so this is something cyclical that happens in the cell all the time. The second way that we have transfer of energy is through redox reactions. So for redox reactions, these are really just the transfer of electrons. So let's go ahead and draw an example of that. So we have A plus B are our reactants, and our products are A plus B minus. So let's go ahead and write up our little cheat sheet. So the one that I like to use is Leo the lion goes grr. So loss of electrons is oxidation. Gain of electrons is reduction. So electrons are negatively charged. This reactant B going to the product B minus gained an electron, and so this is a reduction. And because redox reactions are always paired together, our A losing an electron going to A plus is an oxidation. And so again, we can go ahead and draw our reaction coordinate diagrams for this to remind ourselves of what that looks like for redox reactions, because we'll be drawing a lot of these. So let's go ahead and draw our redox reaction for our A. So A is oxidized, which means it is losing an electron. So because it is losing an electron, it is an exergonic reaction. B is reduced, so we're going from B to B minus, and that is an endergonic reaction. So again, you'll notice that I'm drawing these coupled together. Again, we couple endergonic and exergonic reactions. So keep in mind that even though for our example, A plus B going to A plus B minus, we just used letters. In biology, we don't really count electrons. We tend to leave that to the chemist though. Biologists can be pretty lazy. What we do instead is we look at hydrogens. So remember that hydrogen, if we draw its Lewis dot structure, we draw one dot because it has one electron. So when you gain a hydrogen, you can say that you gain an electron, and so you are being reduced. If you lose a hydrogen, you are being oxidized. So in biology, we're going to look at hydrogens, and you'll actually hear me refer to that a lot. And I think that for biology, for this class, that is what is going to keep us the most sane without having to count all of those electrons. All right, so now that we've kind of summarized topics 13 and 14, now we are ready to move into topic 15. And we're going to start with an overall equation of metabolism of glucose. And this will be a nice way for us to practice our redox reactions as well. So in the metabolism of glucose, we start with C6H12O6 plus 6O2, and that goes to 6CO2 plus 6H2O plus energy. And again, that energy is going to be in the form of chemical energy in those bonds that we are forming, particularly in the bonds of ATP. So let's go ahead and starting with our glucose, I'm going to go ahead and underline our carbons here, and I'm also going to write our little cheat sheet. And let's go ahead and write what is oxidized and what is reduced. So in the case of glucose going to carbon dioxide, glucose starts off with 12 hydrogens. So we said hydrogens are a representation of electrons. It is losing hydrogens as it goes to CO2. And so loss of hydrogens or loss of electrons is an oxidation. So in the metabolism of glucose, we have an oxidation reaction. We also have, you guessed it, a reduction reaction. Because again, those are always paired. That 6O2, when it goes to 6H2O, gains hydrogens. So by gaining hydrogens, it is reduced. So this is our overall equation for metabolism of glucose. And when necessary and as needed, we will go ahead and write out our equations for glycolysis and for some of the other stages as well. And I will come back and remind you of these as we continue. So now we're ready to move into aerobic respiration. So we've got our overall metabolism, and let's go ahead and remind ourselves of what aerobic respiration is. So for aerobic respiration, we've got arrow in the front there. Anytime you see that arrow, that refers to oxygen. And so aerobic respiration is going to be cellular respiration that requires oxygen. We will in the next topic talk about anaerobic respiration, or cellular respiration that occurs in the absence of oxygen. Now, of course, we kind of have a definition here within a definition. So let's go ahead and remind ourselves of what cellular respiration is. Cellular respiration is a metabolic process to convert nutrients to energy. So there are many, many macromolecules that we can use in metabolism to convert those nutrients to energy. In this class, as our equation on the top of this page shows, we are focusing on glucose. And for aerobic respiration, there's 4 stages that we are going to talk about. So hopefully in your notes as you were brainstorming, you jotted down some of the stages. Our first stage is glycolysis. Followed by glycolysis, we have the formation of acetyl-CoA. Stage 3 is the citric acid cycle. And then last but not least, we have electron transport and chemiosmosis. So for the remainder of this topic, what we are going to do is we are going to walk through each of these stages, and we're going to draw what happens. We are going to simplify many of the steps because they're really complex, and if you are really interested in the super complex parts, you will learn them in biochemistry. But for this class, we are building a foundation. So let's go ahead and start with stage 1. So for each of the stages of aerobic respiration, we will return back to this image and we will think about where are we in the process, where is the process occurring, what's going in, and what's coming out. And we will compare these processes in both bacteria and eukaryotes, even though we're mostly focused on eukaryotes. So let's start with glycolysis. So let's remind ourselves of what glycolysis means. So lysis means breaking or splitting. So glyco comes from sugar. So glycolysis refers to sugar splitting. So in glycolysis, we're starting with a 6-carbon molecule, glucose, and that will be broken down. So we're going to write the overall net equation. We're going to color code it here for ourselves, and we're going to draw some energy diagrams as well. So let's start with glucose on the right. It's a 6-carbon molecule, and I'm going to try to do my best here to color code everything. Glucose is going to be converted to 2-pyruvate, which is a 3-carbon molecule. We are also in this reaction going to add 2 NAD+. So remember that NAD+ and then its reduced form, NADH, is an energy management molecule. So we'll see a lot of it throughout this topic. We are also going to add 2 ADP and 2P. Okay, let's go ahead and write our reduced NADH on the right. And we're also going to end up with 2 net ATP and then some other stuff. So we're going to have a little bit of water, and then let me be good and balance my water here. So we'll end up with 2 H2O and 2 H+. So this is our overall net equation for glycolysis. We are going to focus on the colored parts. And so the glucose, the NAD+, and the ADPs. So let's start with glucose going to pyruvate. So when we have glucose that is being broken down to pyruvate, this is being oxidized. This is an exergonic reaction. So we can actually go ahead and draw what that energy coordinate diagram would look like. So there it is for glucose to pyruvate, so an exergonic reaction. And then let's go ahead and make sure that we match our colors. So for NAD plus going to NADH, NADH is reduced. So it's gaining an electron, gaining a hydrogen. That is an endergonic reaction. And of course, when we make ADP plus P and we make ATP, that also is an endergonic reaction. So overall, in glycolysis, we are producing ATP and we are reducing NAD plus to NADH. And so again, you're going to get sick of me saying this, but we are coupling those endergonic reactions with those exergonic reactions. So that energy, that delta G, if we wanted to draw it on to our reaction coordinate diagram for glucose to pyruvate, that energy is being stored in the bond that connects that NAD and that H and that ADP and that third phosphate to make ATP. All right, so now that we have written out our overall net equation, we're going to skip forward to what actually happens in glycolysis. So before break, we took some time to talk about some clinical application or why we care about glucose. So we talked about how glucose is our main source of energy. We talked about some of the hallmarks of cancer, and we mentioned this idea that in different types of cancer, metabolisms are altered, and oftentimes many cancerous cells actually have an increase in glycolysis. In terms of viruses, particularly as we have an outbreak right now of a virus, viruses also affect glycolysis in cells as well. They can upregulate glycolysis to cause an increase of glucose transport into the cell, and many viruses can actually have long-lasting human health effects, such as the hepatitis viruses causing an increase in things like diabetes and liver disease. And so, again, there's many different clinical applications for glycolysis and why we should care. And I'm going to refer you back to our notes from before break, and we're going to hop into actually drawing out glycolysis. So if you recall, glycolysis has two phases. It has the energy investing phase and the energy capture phase. So we're going to take one page per phase and draw out what occurs. So let's start with our energy investing phase. So for our energy investing phase, overall, we have 2 ATP that are invested. So just like we did before break, we are going to represent our molecules just by looking at the number of carbons, and those carbons are going to be represented by circles or by balls. So let's go ahead and draw our 6-carbon molecule, our glucose. So in the first step of glycolysis, our glucose is taken and we add on a phosphate group. So as we mentioned earlier, we reminded ourselves that when it comes to phosphate groups, those phosphate groups are put on by kinases, and that phosphate group comes from an ATP. And I'm going to go ahead and write kinase there to remind ourselves that an enzyme is performing this step. So something to keep in mind is, even though we're not going to be drawing enzymes every single time, enzymes play a role at every single step. All right, so now that we have one phosphate group, we want to even that out. It looks kind of ugly just having one phosphate group on one side of glucose. So we're going to add a second phosphate group to the other side of that glucose. And you probably guessed it, that phosphate group is coming from ATP, and that phosphate group is put on by a kinase. Now at this point, by adding on these two phosphate groups, we have destabilized that glucose. We are in a transition state, and what that allows us to do is to take that glucose and snap it right in half. When we snap that glucose in half, we end up with a molecule that is called G3P. And because our glucose started off with 6 carbons and each G3P has 3 carbons, we end up with 2 G3Ps or 2 glyceraldehyde phosphates. And this is the end of our energy investing phase. So we have gone through, taken our glucose, destabilized it by adding of 2 phosphate groups. Each of those came from a molecule of ATP. Those steps were performed by a kinase. So that is where our energy investing phase came from. And now we are going to talk about phase 2, which is the energy capture phase. So first, we don't want to go through all of that work of putting in energy if we're not going to get anything out of it. So now at this point, this is where we start with those G3Ps. So let's go ahead and redraw those up here. And I'm going to do my best to keep my colors consistent here. And those G3Ps are going to go through a series of several steps. I personally think that this is one of the most boring parts of glycolysis. We go through a series of 5 steps, and in these 5 steps, we have different enzymes that play a role at each of the steps. And we are going to summarize those steps by just talking about what happens during them and not really getting into the nitty-gritty details. If you are interested in the nitty-gritty details, congratulations. When you take Biochemistry 1, you will learn all of them. But for the purposes of this class, we are going to say several steps happen. When we start with that G3P, our final product is pyruvate. So we started with 2 G3P and we end with 2 pyruvates. In this process of taking that G3P and having a bunch of enzymes convert it into pyruvate, 2 things happen. We take NAD plus and it is reduced to NADH, and that's going to happen for each of our G3P molecules. The second thing that happens is we take 2 ADP and those are converted to 2 ATP. And again, that's going to happen for each of our G3P molecules. And so by the end of this process, we end up with 2 NADH and 4 ATP and 2 pyruvate. So if we draw a reaction coordinate diagram for phase 1 and phase 2, it's going to look something like this. And we're going to focus on the exergonic one. I challenge you to go ahead and draw the endergonic reaction for NADH and for ATP. So we start with a 6-carbon molecule. So here's our glucose, and we end with 2 3-carbon molecules or pyruvate. We have this lovely little activation energy, this energy barrier that we have to get over. That activation energy, the energy to get over that, is provided by those 2 ATP that we started with in our first phase. So if you recall from before break, that top of the hump is what we call the transition state. So when we have those phosphate groups on that glucose, that is what is making everything unstable. That's what allows us to go ahead and break that glucose and snap it right in half. So that glucose with those two phosphate groups, that is the unstable part, and that is the transition part of our energy reaction coordinate diagram. So again, this was review from break, but I wanted to make sure that we are all on the same page and that we are all ready to talk about these pieces and to connect them to the next stage. So let's go ahead and summarize what we have said thus far in glycolysis. And we'll actually do this for each of the stages, and we will make sure to keep track of all of our energy management molecules as well. So for glycolysis, this is stage 1 of aerobic respiration, and for the location, this is happening in the cytosol, and that is true for bacteria and for eukaryotes. In terms of what goes in, we have glucose, we have 2 ATP, and for what goes in, you could also be extra thorough. I'm kind of writing down the highlights. We could talk about NAD+ that has to go in, the 2 ADPs that have to go in. I'm kind of writing the main key players down. So if you're wondering why your list has more than my list, that is why. I'm writing down the key players. In terms of what comes out, that glucose is converted to pyruvate. So we get 2 pyruvate at the end of glycolysis. We have 4 ATP. However, we have 2 net, and we end up with 2 NADH molecules. If we were to summarize in one sentence quickly what happens in glycolysis, we could say that glucose is split into 2 pyruvate. All right, we are now ready for a practice problem. So again, I encourage you to go ahead and pause the video and to work through this practice problem as you typically would if we were in class in person. So for this practice problem, we are representing each of the steps of glycolysis as a blue circle, and then we have the first part on the left here. This is the energy investing phase, so we can see ATP is being used up. On the right, this is our energy capture phase. So for this practice problem, I want you all to think about which step in glycolysis is in a redox reaction. So again, go ahead and pause the video, work through the practice problem, and then when I catch you on the other side, we will walk through this problem. Okay, welcome back. So I'm going to go ahead and write up my little cheat sheet. So my Leo the lion goes grr. For some reason, even after, I don't know, it's been over 10 years now in biology, I still have to write it down. And in my head, I still have to walk through loss of electrons is oxidation, gain of electrons is reduction. Maybe in another 10 years that'll change, but we'll see. So we're going to go ahead and focus in here on this NAD+ and this NADH. So whenever we have a loss of electrons or a gain of electrons in biology, we're looking at what happens to that hydrogen. So out of all these reactions, we only have one hydrogen here that is present. So we see an NAD+ going to an NADH. That NAD+ is gaining an electron, and so that means that it is a reduction. So here is our reduction, and that is happening at reaction D, or step D, if you will. And we know that redox reactions come in pairs. So where there is a reduction, there must be an oxidation. Therefore, step D is where redox is occurring in glycolysis. So let me know in the comments below if you got this practice problem. If you are lost or if you are confused and you want to go through it again, feel free to contact me and we will do that. Okay, so now that we've done a practice problem, we've summarized glycolysis, now we're ready for stage 2. So stage 2 is the formation of acetyl-CoA. So we're going to draw both a simple model and then we'll actually draw out the molecular model. You don't have to know how to draw the molecules. This is more to just show you and explain where some of the names come from. So let's go ahead and first talk about where this is happening. So the pyruvate from glycolysis has to enter the mitochondria. Now keep in mind, this is just for eukaryotes because our prokaryotic friends do not have mitochondria. So in eukaryotic cells, that pyruvate that we just made, that we ended with in stage 1, has to be shuffled into the mitochondria. In prokaryotes, in bacteria, archaea, this process continues happening in the cytosol. So we're going to introduce a new enzyme in this stage. The enzyme that we're going to talk about is pyruvate dehydrogenase, kinase when enzyme names make sense. And what this enzyme does is it's going to catalyze the reaction that takes pyruvate to acetyl-CoA. So again, let's go ahead and let's draw a model of what is happening. Right now, we've kind of just said what happens in words. So on the left, we'll draw a simple model. So we're going to keep with our circles representing carbons. So here is our pyruvate, a 3-carbon molecule. This pyruvate is going to be converted to a 2-carbon molecule that has a CoA or a coenzyme A attached to it. So during this process, we're going to draw our little enzyme. So our little Pac-Man onto there. So our pyruvate dehydrogenase. The enzyme takes pyruvate and it's going to take CoA and it's going to knock off a carbon off of pyruvate. That carbon actually leaves as CO2, and it's going to attach CoA. While this entire process is happening, we actually get pretty lucky. We actually make an energy management molecule here. So while this is occurring, we take NAD+, and NAD+ is reduced to NADH. Because we have a reduction, that means we also have an oxidation. Because we have an oxidation and a reduction, and you and I are really smart, we can go ahead and draw our reaction coordinate diagrams. So let's go ahead and do that. So let's do it first, since I'm in blue, let's do it for NAD+ going to NADH. So when you are reduced, you are gaining an electron, and a reduction is an endergonic reaction. So this is our endergonic reaction. Because we have a reduction and an endergonic reaction, you and I know those are coupled to exergonic reactions. And the only other thing that is going on right now is we are taking pyruvate and converting it to acetyl-CoA. So what that means is that that must be an exergonic reaction. So here's our pyruvate and then our acetyl-CoA at the end of that step. So we've got our oxidized and our reduced, so our redox reaction that occurred. So again, this is our simple model, and I am going to go ahead and highlight the CO2. And we will come back to the CO2 and a modeling activity that we're going to do in about a week or so in this class. But keep track of those CO2s. They're going to become really important. So let's go ahead and kind of block off that. And let's draw what pyruvate looks like, and let's draw what acetyl-CoA looks like. It will help us learn a new functional group, so what an acetyl group is, and it'll kind of make a lot more sense as to why this thing is called acetyl-CoA. So for pyruvate, it's a 3-carbon molecule. Let's go ahead. All right, so here we go. So here is our pyruvate, and we're essentially going to draw our simple model that we drew on the left. We're going to redraw it on the right, but we're going to draw it with the molecular structures. And I'm going to kind of draw it diagonally because I did not leave myself enough space here. So when we look at what is going on, we know from our simple model that one of the things that comes out of this reaction is CO2. I'm going to draw a dashed circle-ish lines around where that CO2 comes from. So that CO2 comes from the right side of that pyruvate molecule. All right, while this is happening, and let me keep my color coding the same here, we're going to have that reduction of our NAD+. Let's go ahead and add our enzyme in. That enzyme, when it is finished, we will end up with our CoA that is attached to 2 carbons that are left over from pyruvate. This part of the molecule that is circled in that light green turquoise color, this is called an acetyl group. So we've seen a lot of different functional groups throughout this semester. This is just a new functional group. So the reason that we call this acetyl-CoA is because the coenzyme A has an acetyl group that is attached to it. So we have gone from 1 6-carbon molecule to 2 3-carbon molecules known as pyruvate. Each of those pyruvates has now been converted to a 2-carbon molecule with a CoA. And so at the end of this, we have 2 acetyl-CoAs, we have 2 NADH molecules, and we have 2 carbon dioxide molecules. All right, so one more thing that we're going to talk about for this stage before we summarize it, lucky for us it's a pretty short stage, is we're going to talk about a deficiency called pyruvate dehydrogenase deficiency. So although metabolic disorders are rare, here is an example of a metabolic disorder. And so this is a neurodegenerative disorder. It is X-linked. So it's a genetic disorder, and it's a problem with a mitochondrial protein. So overall, when we are in the mitochondria, so let's go ahead and remind ourselves of what that looks like. So there's our double membrane. All of our enzymes that are working on the stages that occur in the mitochondria are present in the matrix. So that's this inner part here, or in the inner membrane. So for pyruvate dehydrogenase, this is an enzyme that is found in the mitochondria. So when we started stage 2, we talked about how that pyruvate had to be imported into the mitochondria in regards to eukaryotes. So we are in that matrix, and in pyruvate dehydrogenase deficiency, as the name implies, we have a problem with our pyruvate dehydrogenase. So one of the things that happens, as the image on the bottom right shows, is we get abnormal brain development. So on the right, we have normal, and on the left we have what we refer to as microcephaly. So microcephaly is a smaller than normal head and smaller than normal brain as well. And for these patients, they typically will die before the age of 10. And in terms of other statistics, we have seen out of about 240 patients that have pyruvate dehydrogenase deficiency, about 10 of them are able to live until they're 20. So in terms of treatment, what do we have? There's not really a lot that we have for treatment. Maybe in a couple decades or so, we'll be able to fix their gene and to give them a good copy of pyruvate dehydrogenase. But for now, the best thing that we have for patients is we encourage them to try to keep a low-carb diet. And so we don't know too much about this disease. It's pretty understudied, but it is a disease that is very relevant to what we are talking about. So let me back up for a second. So here in our simple and our complex model on the right, we have drawn pyruvate dehydrogenase, and it is the enzyme that takes pyruvate and breaks it down. So you can imagine if you have an enzyme that is broken at this step, you stop at this stage. You stop at pyruvate. You never continue forward because your pyruvate dehydrogenase is unable to break that down. And so what ends up happening is you can have a lot of waste and byproducts. And so we don't have enough studies, but we do have some data that suggests keeping a low-carb diet or keeping a ketogenic diet helps these patients a lot. So again, this is a very understudied disease, but it is really important to what we're talking about, and I promised you all I would do my best to try to bring in medicine and tie in relevant examples as best as I can, and this is a disease that I found that affects this stage. So let's go ahead and do a practice problem. All right, so hopefully you have paused the video and you have walked through this problem. So let's start with just reminding ourselves of what's going on. So here is our pyruvate, so our 3-carbon molecule. Our pyruvate is converted to acetyl-CoA, and in that process we make NADH. So I'm going to go ahead and write my cheat sheet up here. And let's walk through these together. So let's start with D. So D says that CO2 is consumed. We can cross that out because in this process, when we take one of those carbons from pyruvate, we take a carbon and 2 oxygens, we actually produce CO2. So that is false because CO2 is produced. For C, ATP is produced. Again, we don't have any ATP. In this step, we're lucky that we even made an NADH. So we can go ahead and cross that off. The next thing that we have is NAD plus is oxidized. So in this reaction, NAD plus goes to NADH. So we have gaining of a hydrogen, which is gaining of an electron, which is a reduction. So this again is false because NAD plus is reduced. So now let's take a look at pyruvate. So we said that if NAD plus is reduced, then the other partners must be oxidized. So pyruvate to acetyl-CoA is oxidized. So that one is true. Again, if you have struggled with this problem, feel free to, during our next office hours, we can do any sort of virtual office hours. We can walk through this together. So let's go ahead and summarize this stage. So for the stage of aerobic respiration, this is stage 2. For our location, depends on what type of organism you are. If you are a eukaryote, this is occurring in the matrix of the mitochondria. If you are a bacteria, this is occurring still in your cytosol as you have no mitochondria. In terms of what goes in, we have 2 pyruvate And what comes out? We have 2 CO2, 2 NADH, and 2 acetyl-CoA. And finally, if we were to summarize a general one-sentence statement of what is happening, pyruvate is converted to acetyl-CoA. Okay, so now that we have gone through more than one stage, we are actually going to summarize our energy management molecules that we have gained so far. So for glycolysis, let's start with ATP. We have 2 ATP, and that's our net. We had 4, but we had to use 2, so net we only have 2. We also produce 2 NADH in glycolysis. During the production of acetyl-CoA, we had 0 ATP and we had 2 NADH. So 1 NADH was reduced for every molecule of pyruvate that was oxidized. So total, so far, we are at 2 ATP and 4 NADH. So not too shabby. But we will see as we continue through the later stages that is where the majority of our energy conversion or energy management molecules come from. Goodness, you can tell it's getting near the end of the lecture and that I am getting tongue-tied. Next thing that we're going to do is we're actually going to stop the video here, and what I've decided to do is for some of these longer topics, rather than having a 2-hour long video, is to split it right down in the middle. So this is the end of topic 15, day 2, since we technically did day 1 before break. So the second online class will be topic 15, day 3. And for that, we will talk about the citric acid cycle and the electron transport chain. So thank you for making it through the first online lecture with me. Hopefully these get easier the more that I film them. I will say it's been a little bit of a struggle. You all will have to let me know if obviously I'm not there in person, but if this is at least somewhat reminiscent of what our typical lectures were in person. So I hope y'all are doing well. Stay safe. I miss you, and I will see you soon. Have a wonderful rest of your day. Thanks. Bye.